In today's video, we'll be discussing blood composition, red blood cell breakdown, hemostasis, and a couple of other things, but excuse me. In this first portion, we'll be talking about the components of blood. So blood is composed of about 55% plasma, that's shown here. Ooh. Okay, let's zoom in. Sorry. 55% plasma, and then other elements such as red blood cells, white blood cells, and platelets. Okay. Plasma is composed of basically, again, 55% of a person's blood, but overall, it's 92% water, about 90% water, but the other portion is a bunch of proteins. We have albumin, which is the most abundant fibrinogen, and globulin. Okay. Next, we'll be talking about basically how red blood cells are broken down. So we have hemoglobin. Let me just write this up. Hemoglobin. Heme portion will be taking this pathway. Globin is just broken down into amino acids. Oh my goodness, I can't spell. Okay, so the heme takes a different pathway and turns it into bilirubin. So basically what happens, you have hemooxygenase, which converts it to biliverdin. Biliverdin reductase then converts it into bilirubin here. Okay. And then I kind of want to talk about just how bilirubin is, what's it converted to and how it kind of goes out throughout the body. So basically, bilirubin's going to come in here into the liver. And I wrote this out here if you want to take a photo, you can take a picture out of it or whatever. So bilirubin comes into the liver, is transferred by albumin. Once it's in the liver, it becomes conjugated through glucuronic acid here. Then bilirubin, the conjugated bilirubin, is going to come through bile duct. It's going to be pushed into the intestines here. And it's going to be converted into the bacteria in the gut. It's going to convert it to I believe. It's going to turn into urobilirubin or urobilinogen. And some of that is going to return back into the blood. Some of it is going to actually go towards the kidney and become urobilin, which is basically your urine. What else do we hit? We hit red blood. Some of it's just recycled. It just enters this enterohepatic urobilin cycle. And then the other portion that's left into the intestine is converted into stercobilin, which is basically what gives your stool the dark brown color. So yeah, that's that whole process. Now we're going to talk about the different mechanisms that contribute to hemostasis. So the first really easy one is vasoconstriction. Vasoconstriction basically constricts blood vessels, which then ultimately reduces blood flow. Okay, and then we'll talk about platelet aggregation, coagulation. Okay, so this is a little confusing, gets kind of complicated, but ultimately we have platelets here. And these platelets are just in the blood. And if there is a rupture or a breaking of the endothelial cells, VWH will be released, which then binds to VWH receptors, and that allows it to bind to the collagen here, right? And then as blood is moving, it basically activates this platelet cell through a mechanical process. So this is the activated platelet cell, and once those suckers are activated, they'll release a variety of different things based on the granules. So alpha granules will release more VWF, which again activates them. Factor VIII, which we'll talk about later, and platelet-derived growth hormone, which basically just tells skin cells to grow. And then dense granules will release calcium, and calcium basically just allows for further activation and ADP as well. They are activated, which then they will again continue to release fibrinogen or fibrin. Right, yeah, fibrinogen or fibrin, I can't remember. I believe it's fibrin. Apologize. Which then those suckers also release TXA2 or thrombin. Those activate more platelets as seen here, which then allows more platelets to eventually bind to one another through GPS2B3A, which is a fibrinogen, and they form bridges between all the platelets, allowing them to stick together. And this all kind of happens pretty quick, and it seals up that area here. Okay, now we'll move on to the coagulation cascade. This isn't super confusing. Basically, what happens with you have your Hagman factor, 12, and that's going to bind to these negative charges on the collagen and turn it into 12A, and then 11, factor 11 is going to come in, turn it into factor 11A through factor 12A, and this will keep going on and on until we basically get to the most important portion, which is when prothrombin turns into thrombin and fibrinogen turns into fibrin. And fibrin is what makes the mesh on the hole, basically. And that whole portion was part of the intrinsic pathway and common pathway. Intrinsic means in, so that means it's activated within. Extrinsic means there's something outside that's activating it. So the cells are actually ones that have this factor 3, which is called a tissue factor. So factor 11, which is made by the liver, comes in. It's activated by factor 3, which makes factor 3A. X comes in, activates, is activated by factor 3A and becomes 10A, which then is able to come all the way over here into the intrinsic, or common pathway, and allow for the further conversion of prothrombin to thrombin. Yeah, and that just allows for coagulation and coagulation. And then we'll talk a little bit further about plasminogen and plasmin. So plasminogen is just a protein that's in the blood. It's just a zymogen that's just hanging out. And ultimately, we want plasmin, which allows for what's it called? I'm trying to find the right word. Let's see. Fibrinolysis, which is like the regulation of fibrin, and it just kind of breaks down the blood clot so that it doesn't continue further down the bloodstream and cause an embolism. And so endothelial cells with fibrin will actually release what's called TPA, and TPA then activates or converts plasminogen into plasmin. TPA basically just elevates this conversion. And yeah, like I just said before, plasmin then breaks down fibrinogen through a process called fibrinolysis, and that just breaks down the clot and allows for, so it doesn't allow for an embolism. yeah, that's basically all we have today on blood and coagulation and all that stuff.